We will record this for posterity. If you don't want your beautiful face shown, please turn your camera off. So today we've got an event. We've got the first joint APGC IBH seminar. And it's good to see young blood talking as well. We've got Lorna McAusland, who's at Nottingham University. Lorna's a plant physiologist, and she's specialising in the dynamic responses of photosynthesis and water regulation to heat stress. Climate change is a biggie, and I suspect all of us are working on it in one shape or form. So this is a very pertinent talk. I'm not going to talk about the seminar synopsis because I'll get it wrong. You should have read that, but we'll hear the talk itself. That's what we want to hear. So Lorna, it's over to you.
Wonderful, thank you for that introduction. I'm just going to share my screen and hopefully that will all go smoothly. Right, can everybody see my screen? Is it all there?
Kind of skewed to the one side, the way I'm seeing it.
Yeah, the left-hand side is cut off for some reason.
Yeah, I also have that as well. Don't believe how many of these I've done and they still don't get smoother. Like, how many years have I been doing online talk? Is that working for everyone?
That's it.
Perfect.
Wonderful.
That's it.
Wonderful. So thank you very much for that introduction. My name is Lorna McAusland. I currently work in Professor Erik Murchie's lab, but recently I was awarded a BBSRC Discovery Fellowship. So today what I'm going to do is I'm going to talk about the work that I completed as part of a BBSRC-Newton project. So I'll describe what that is and some of our main results and exciting things going forward. And then I'll talk a little bit about my fellowship work going forward as well, so you have some background. So the title of my talk today is Things That Go Bump in the Night, Understanding the Role of Nocturnal Conductance in Response to Climate Change. So, the outline of my talk. Why worry about nocturnal processes at all? What plant processes actually occur at night? What is nocturnal stomatal conductance? Does it occur in our modern wheat genotypes? Is there underlying genetic variation for this trait? Do we observe these responses in the field? Because most of the work that we can do is done in growth rooms in the UK. And then finally, what are the implications of nocturnal conductance in wheat? So nocturnal temperatures is where I'm going to start, and you're very right, it relates all the way back to climate change. When we think of heat waves, we tend to think of long, hot, sunny days, and that's what we think of as a heat treatment. But actually, nocturnal temperatures are rising about 1.4 times the rate of daytime temperatures. And this is worldwide in very specific areas. So if you look at this map, this is from Cox et al. In 2020. The blue region of the map is where temperatures are rising faster at night. The red areas of the map is where temperature is rising faster at the day. So you can see in Australia, you're getting more increases in temperature a little bit at night, but mostly in the day. In the UK, you've got rising temperatures at night. And also in places like the sub-Saharan Africa, you have increases at night. And why is this? It's mostly down to cloud cover. So increasing daytime temperatures as well cause increases in evapotranspiration. You get dense cloud cover over particular areas of the world. That dense cloud cover keeps the heat close to the surface of the Earth, and that means we get warmer nights. The air is trapped and it's warming up. So why is this a problem? So in 2007, Lobel and Ortiz Monasterio discovered that for every 1 degree increase in minimum temperature or nighttime temperature, you get about a 10% decline in spring wheat yields. And this isn't just in wheat, it also happens in rice and in barley. And it's only now that we're really starting to explore phenotypes that determine characteristics which enable nocturnal heat tolerance, not simply responses to daytime heat stresses. So what happens at night? Well, the environment is usually a lot cooler at night because you don't have the sunlight. And you normally get in areas where you have high daytime temperatures and very cool nighttime temperatures, you have something called temperature inversion, which means that you get a warm layer of air that's warm and sitting on top of a cold layer. These two layers don't mix, and that means that you get much stiller nighttime air, which means you get greater boundary layer over objects such as crops. And that means you get less evaporation, so you get higher humidities. If you have warm daytimes and then suddenly cold nighttimes, you might get dew falling on your leaves, so water droplets. You might also get low soil water evaporation, and you can have something called hydraulic redistribution, so water moving from abundance in the soil into areas that are drier, for example, the areas around roots. What about in the plants? So obviously during the night we have no photosynthesis, and instead we have dark respiration. We have something called folio uptake in some species, and that's when you get high areas of humidity or water on the leaves that's taken into the leaves. And this is very common in trees, especially very tall, very old trees. You can have a trait called circadian nyctinasty. If anybody's bought a prayer plant over lockdown and you see the leaves moving up and down, that's circadian nyctinasty. It's when movement's induced by the change of day to night, and it's normally because of changes in water potential. And you have something called nocturnal stomatal conductance, which I'm going to focus on for the majority of this talk. So stomata exist on almost all areas or all aerial areas of the plant. Mostly we look at them in leaves. You can see two stomata here. They're guarded by two guard cells, and they regulate the openings of the leaf, and they allow the exchange of CO2 for photosynthesis and water lost via transpiration. And up until recently, the main thing we would talk about when we talk about stomatal conductance is response to light. So increasing light increases stomatal conductance, also higher humidity or low CO2. However, recent work has shown that stomata will also open at night, and this is what we call nocturnal stomatal conductance. And it's not a small amount. So nocturnal stomatal conductance can account for up to 30% of daytime rates of water loss. But why is this a problem? So in 2017, Lombardozzi used the data for nocturnal stomatal conductance and said if there was a 5% increase in nocturnal stomatal conductance, there could be up to a 50% decrease in available soil water in semi-arid regions. And this is a big deal because most of the models that we use to predict how climate change will affect areas of the world, so not just crops, but for example, rainforests, etc., they don't take into account water loss at night in this particular way. So this is really important and could have really big implications for the water cycling at night, and especially in response to climate change. So there are a couple of really basic questions that we had for this project. Simply, does nocturnal conductance occur in wheat, in a crop plant? And is the magnitude of nocturnal conductance genotype specific? Is there genetic variation underpinning nocturnal conductance? So this project is centered on wheat, and I don't know how much everybody in the audience knows about wheat. It is our most grown crop globally on an area basis. In fact, 60% of the wheat that we have in the world is grown by 6 countries. The top of that is China. So last year they produced 135 million tons. That's about 18% of the world's supply. If you're interested, the UK produced 15 million tons. So it's about 12th on that list. And then the country I'm going to talk about now, which is Mexico, is 21st on that list, and it produced about 3.5 million tons. So the project I'm on is a BBSRC-Newton, which means it funds the collaboration between a UK university or institute with another country. In this case, it is Mexico. Most of the wheat in Mexico is produced in the northwestern corner in 2 states, Sonora and Chihuahua. So you can see here on this map, there's a very, very dark green area around Ciudad Obregón, or Obregón City. So Obregón is the home of the Wheat Physiology Hub, which is run by CIMMYT, the Centre for Maize and Wheat Improvement. CIMMYT was responsible for the release of about 950 different cultivars to over 60 different countries since 2012 to try and combat yield stability under climatic change and economic uncertainty. So they have a real role to play in what materials released worldwide and what's grown worldwide. And most of the material is assessed here in Obregon. This is also where Norman Borlaug did most of his trials when he won the Nobel Peace Prize. So if we look back at our map, we can see that Mexico also has this problem of high nighttime temperatures. And although the resolution on this map isn't great, it is also mostly down to cloud cover in that particular area of the world. This is what a field in Mexico looks like. Wheat as far as the eye can see, and then you can see the mountains in the distance. The wheat fields actually border a desert. So here's the view from the plane as you touch down, the Sonoran Desert on one side and the wheat on the other. 80% of the wheat in Mexico is irrigated. And from this particular field site, it's irrigated by the El Vero Dam, which supplies about 450,000 hectares with water. What's the climate like? So you can get maximums of about 47 degrees in the summer and minimums of about -7 in January. The growing period is December all the way until April, May. And these are extremes. But what I want to highlight here is the difference between the minimum temperatures and the maximum temperatures is large. In Nottingham, the standard difference between your minimum and maximum temperatures is about 8 to 10 degrees. In Obregon, it's between 15 to 17 degrees. So the variation is quite big. So the climate at this particular CIMMIT experimental field site is the perfect condition for assessing the yield potential. So what maximum yield we can get from high sunshine, well-watered, nutrient-fed crop. But it's also the perfect place to assess adverse environmental conditions. For example, we can withhold water to assess drought, or for example, sow later to assess the impact of heat. Obviously, most of the year I am not in Mexico. I normally go there for a couple of months each year, and it's been about 6 years of doing that. But we have these facilities at the University of Nottingham that enable us to replicate weather. So here we can replicate temperature, humidity, and light in these growth rooms. So here you've got about 1,000 plants, so that's about 250 lines with 5 reps. And they're growing in a simulated season in Mexico. So you can change the maximum and minimum temperatures and the light curve. Also very handily for me, as this is a nocturnal project, you can shift the dawn so you don't have to come in at really antisocial times. This is some data from that room again, and you can see our plants looking very happy, just tillering away there. So what did we do first? So first we did grow some plants in that growth room in Nottingham, and what we picked was 6 different Mexican spring wheat lines and 2 land races. And if you're not sure what a land race is, it's kind of the intermediary between a wild relative that you wouldn't eat and an elite variety that we use today for making bread wheat or pasta. And what we did was we did some gas exchange on them, which means we assessed the amount of water lost via stomatal conductance on the left and CO2 uptake as photosynthesis on the right. And we saw that there were significant differences in those between our genotypes. If we then looked at night, we saw that there was significant difference in respiration as well, so the amount of CO2 released at night, and significant differences in nocturnal stomatal conductance. And there was actually a 3-fold difference in mean nocturnal conductance for our wheat genotypes. In particular, there was this line, aptly named Borlaug 100, which is an elite Mexican spring variety. It was over almost 20% of the daytime rates of water loss just at night. If we split this data down further, so we split it by growth stages, so each of these boxes is a genotype, and then each bar is a growth stage going from young material to old, you can see that we had very different responses for our genotypes. Borlaug managed to maintain high nocturnal conductance throughout most of its life, whereas something like, I don't know, Vorobe, which is down on the bottom there, it had high nocturnal conductance only really in its tillering stages or its pre-flag leaf stages. This is a lot more obvious if you look during the day. The magnitudes are much, much higher in stomatal conductance during the day. that most plant material had higher rates of water loss when it was younger than when it was older. So we also found some nice relationships. So the first relationship, which is quite an obvious one as a physiologist, is that if you had higher daytime rates of stomatal conductance, generally you had higher rates of CO2 assimilation. Stomata are enabling that uptake of CO2. The wider they are open, the more CO2 they take up and are able to fix. But we also got a nice correlation between daytime rates of water loss and nighttime rates of water loss. The more water you lost during the day, the chances are you had higher nocturnal conductance as well. And we also saw this relationship. So there was a negative relationship between respiration and nocturnal conductance, i.e. if there was higher amounts of respiration with higher amounts of nocturnal conductance. One of the things we stopped to assess here was cuticular conductance, because anybody that has done any kind of gas exchange in the field or otherwise knows that you want to make sure that you're measuring an active process. You aren't just measuring a damp leaf. And nocturnal conductance is one of those things where the magnitude of conductance is much, much lower than daytime conductance. And one of the questions I got very early on in the project is, how do you know that you're not measuring cuticular conductance? So cuticular conductance is when the leaf is growing, and basically you have what I would term, very unscientifically, a leaky leaf. So it's growing, there are gaps, it's releasing water, and this is a passive process, not an active one where the stomata would be controlling it. But fortunately, when we looked at cuticular conductance, it was an order of magnitude lower than nocturnal conductance. Interestingly, it was also genotype specific, and it was growth stage specific as well, but it wasn't the same magnitude as nocturnal conductance. Being at heart a stomatal physiologist, I also looked at the anatomy, and we found that nocturnal conductance didn't seem to be dependent on stomatal density or the ratio, so the number of stomata at the top of the leaf or the bottom of the leaf. However, it was surface specific. So at night, the stomata were more open on the top of the leaf than they were on the bottom, consistently. And this is really exciting to me because it suggests that different surfaces of the leaf have different amounts of control dependent on the environmental things they're exposed to. So lastly, we found that nocturnal conductance was positively correlated with harvest index, so the seed to biomass ratio. And that correlation was only found in the later material, so the older material, suggesting to us that nocturnal conductance isn't just a wasteful water process that happens at night, but there might be something to do with it being open at night and relating to seed yield. So then we went out to the field. So we took all of this data and went out to the field, and there's a little bit of background. We had 12 different genotypes that we grew, and they had differences in heat tolerance. When we say they have differences in heat tolerance, this is an assessment of the decrease in yield in response to a late sowing. So a sowing that was sown later in the season, so you get a decrease in grain number, for example. So these genotypes had variations in heat tolerance, so generic heat tolerance, and this is done in the field. And this fantastic setup was produced by ITSM, so the Institute for Technology in Sonora, and they based it on a publication by Bruce Kimball. And what you can see in the field are these infrared heaters and environmental loggers. So we had 4 plots, 2 that were control and 2 that were heated at night. The sensors worked on a feedback regime. There were sensors pointing at the controls and sensors pointing at the heated plots. And whenever the heated plots fell to 2 degrees lower than the controls, the heaters turned on. So they did this throughout the night. They'd turn on and turn off again when they reached temperature. And there were numerous temperature sensors in the field. We also had different temperature sensors, different sensors in general. So we have a leaf wetness sensor there that I've got a photo of, because Mexico experiences heavy dew. And by heavy, I mean about 2 hours after sunset. If you've had a clear day, you will be soaked. It's very, very heavy dew out there. We also had these about 1.2 meters above crop height, and they were adjusted as the plants grew as well. Occasionally, we would get a bit close to them. We were all a bit aware of having the kind of smell of burning hair occasionally, and because they are quite close to you when you're standing in the field and you're the same height as me. So this is a picture of us at night. So you can see the glow from Obregon, a little bit of light pollution there and a very sensitive iPhone camera. But you can also see the glow of the IR heaters turning on. You can also see some of the guys from Summit. They're wheeling a phenomobile through the plots and they're taking thermal images, but also spectral indices as well. So did it work? So we used IR guns to assess whether it worked, and here is a schematic of the field. So the top two are our two biological reps that were in the control. Each rectangle within them is a genotype. And then the bottom two are our heated plots. Each rectangle is a genotype. And you can see that our heated plots, by the warmer colour, so the more red colour, are actually much, much warmer, 2 degrees warmer than our control plots. So we also did some gas exchange, and I want to show you the initial field trial results because I presented some of this work in Monogram in May last year, I think. And these are the results I presented, but I wanted to show you the difference between the results I presented there and the results also that I found by the end of this year, basically. So initially we found that there was very little difference in daytime stomatal conductance between our control and our heated, either at booting, so the younger material, or heading plus 12. So that's heading, so when the spike emerges, plus 12 days. But at night, nocturnal conductance was significantly affected by heat. So nocturnal conductance decreased in response to heat. If we look during the day, there was no significant effect of heating. So the plots that were heated did not fix more carbon, nor did they lose more carbon. However, dark respiration decreased in the heated plots. The respiration became less negative. And this was the same whether you were younger material or older material. How did this look when you look at the field? This is a false colour image. Again, you've got your two plots at the top are your controls, and the two plots at the bottom are your nocturnal heated plots. Each plot within them is a genotype. The more blue the plot, the higher the conductance, the nocturnal conductance. And these are our two genotypes that we're quite interested in. So Ciano, which is a durum wheat, and then we also have Borlog 100 again, which is our Mexican spring wheat line. And interestingly, when we take those results in the day, we also find that these plots had high stomatal conductance. And this is really exciting for us because it notes that potentially, if we have high daytime conductance, we also have high nighttime conductance, which is a good indication of things going forward. However, were they consistent? So this project's been running for 3 years now. We're in the 4th year after an extension from the ODA funds. And we found that CO2 assimilation did not respond to nocturnal heat still. So that's consistent with the initial results. Here you have a graph where you have booting on the left, so our younger material, and heading on the right. The blue plots are your control, the red ones are your heated. And stomatal conductance didn't change. There was some shift in 2020, but in general 2020 was a lot warmer year than 2021 or 2022. So in general the effect was probably felt more. Now at night, we did see that respiration declined again. It declined in response to heating, which is potentially opposite to what we might think. You might think that respiration would increase under more stressful conditions, but it didn't. And one of the key findings was if we looked at the respiration to assimilation ratio, so the amount of CO2 released versus the CO2 taken up, we found that the greater declines in this ratio indicate acclimations. This is a well-known thing that if there's a decline in the ratio, it indicates better acclimation to the conditions. So our high tolerant lines, some of the lines that we had in the field, indicated greater declines in booting than in heading. This suggested that there was a higher degree of acclimation in our younger material, and that potentially would play a greater role in heat tolerance under severe temperature stress. So for example, you have the two on top here, so minus 40% and 44%. Those two genotypes are our most heat tolerant genotypes, and they have the greatest change in the ratio of respiration to assimilation. We also have our genotype ending 8121 that had no change whatsoever in response to the nocturnal heating. Now, what about our stomatal conductance? There was a consistent decline in our stomatal conductance in response to nocturnal heat for every single year. So, in a summary, nocturnal conductance in the field was genotype specific. It was greater on the adaxial surface again, so on the top surface was higher than the bottom, which is what we saw in the growth room. It declined with age, but increased in contribution. So, as I said before, stomatal conductance declines with age, whether it's daytime or nighttime. Daytime conductance declines much more rapidly than nighttime conductance, which means that nighttime conductance makes up a greater proportion of the water lost as the plant ages, just due to that fact, that difference in magnitude. And it declined with high nighttime temperatures. However, when we look at that lovely relationship between daytime stomatal conductance and nighttime stomatal conductance, it was only weakly correlated. And when we heated the plants, that relationship between water lost during the day and water lost at night completely vanished. So that artifact that we saw in the growth room, we just didn't see it in the field. So what about our grain yield? So overall, our grain yield decreased by almost 4% in response to heating. Our 1,000 grain weight didn't really change. Our harvest index went up, mainly as a result of our biomass going down more than our grain yield. So that declined by almost 6%. However, if we split the data only by tolerance, so if we just split the genotypes into whether we considered them a high heat tolerance line or a low heat tolerance line, we found that our high temperature tolerant lines were the only group to show significant decreases in grain yield in response to high nighttime temperature. So our intermediate and our low tolerant lines had 36% and 22% higher grain yields under nighttime temperatures than our high temperature tolerant lines, which we thought was very surprising. Now, I like to put this slide in because sometimes like 3% doesn't sound like very much in terms of difference. So a 3.83% reduction in grain yield is not very much. If we think back to the Lobel and Ortiz Monasterio paper, they said 10% for every 1 degree increase in T-min. So we got about 2% for every 1 degree increase in T-min. So almost 2% decrease on 3.5 million tonnes is about 66,500 tonnes of grain. And that's the same amount of grain North Korea produced in entirety in 2022. So the implication is there that even small changes or small responses to nighttime temperature are having large impacts on our grain yield for wheat worldwide. So in summary, there's a lot more work to be done. There always is when you get to this part of the project. We know that nocturnal conductance occurs in wheat, but it's genotype specific, it's growth stage dependent, and it's reduced under higher nocturnal temperatures. However, we're still exploring that mechanistic role for nocturnal conductance. Is there a yield penalty for low or high nocturnal conductance? Is it related to carbohydrate translocation? So we did look at carbon and nitrogen flux, and it wasn't significant in our heated plots. And does growth stage specific acclimation play a role in nocturnal heat tolerance? And is it linked with that magnitude of nocturnal conductance in those genotypes? And most interestingly, that nocturnal heat tolerance may be at odds with daytime heat tolerance. I'm not saying that one is more important than the other, and obviously if you have a combination of nighttime and daytime heat stress, that's going to have an overall highly negative impact on your yield. But the mechanisms that enable nocturnal heat tolerance have had much less attention than the mechanisms that enable daytime, and whether nighttime could act as a buffer against daytime heat tolerance, etc. So there is some future work to be done. There is a field season now going ahead, and hopefully I'll be out there again at the end of March. So one of the questions I get asked quite regularly when I give this talk is that heat is often accompanied by drought. They often come hand in hand, and it's very difficult sometimes to separate them. And in fact, heat and drought combined, they account for about 40% of the variability globally in yield. And by the end of the century, it will be about 60% of the variability in wheat yield. So we wanted to address this and to come up with some more data and to hopefully extend the project. So instead of having our 4 plots where we had 2 heated and 2 control, we are now doing 4 treatments. So one will be a control plot with irrigation. We'll have a nocturnally heated plot with irrigation, but we'll also have a control plot with drought and a nocturnally heated plot with drought. Those poor plants are going to seem quite crispy by the end of it, but you need all 4 of them to really tease apart those genotype-specific differences and see what's going on. So now I'm going to go through my acknowledgements for this project, and then I'm going to move on to summarising my BBSRC discovery work going forward. So first off, thank you to the University of Nottingham. The PI on this project is Professor Erik Murchie, and I've had some fantastic help from, for example, Darren Wells and Jonathan Atkinson during this project to really set up some really exciting things to happen at night. The Earnham Institute, we currently have an RNA-seq experiment running for nocturnal conductance on key lines from this project. ITSIN, so the Institute of Technology in Sonora, specifically Drs. Enrico Yepes and Dr. Jaime Garatuza-Payan and Dr. Suzuki Pinto. They were fantastic on the ground. They knew exactly where to go. They were doing all of the infrastructure, and they were fantastic help with the measurements because field work needs a lot of help to get off the ground. And then finally, thank you to CIMIT. So Dr. Matthew Reynolds runs the Wheat Physiology Hub in Obregon, but I had fantastic amounts of help from Dr. Liana Acevedo-Siaka, Dr. Francisco Pinto, and Dr. Hema Malero. So this is a really quick overview of my BBSRC Discovery Fellowship. So hold your questions for nocturnal conductance, and if you have any others on this, I'm very happy to answer them. So my Discovery Fellowship was funded in December last year. It was a very nice Christmas present to me. And it's entitled Exploiting the Untapped Potential of Non-Foliar Photosynthesis in a Warming World. And the astute of you will notice there in the picture, I have a lot of wheat spikes and also wild relative spikes. So when most of us think about where photosynthesis occurs, we think about the leaves, but it also occurs on all other green aspects of the plant. So for example, the fruits, the stems, and even some flowers. Now, arguably the most important flower there is the wheat spike. This is a film I took in Mexico, and you can see it being buffeted in this environment of high light and high heat. And it really struck me as a part of the plant that we see, but have very little ability to measure in terms of photosynthesis. It's very complex and dynamic. So the wheat spike itself changes shape, not only in response to abiotic stress, but also day to day. So on the left, you have a time-lapse video I took of a desiccating wheat ear, and on the right, you have a diurnal movement and flowering motion of a wheat spike. So a wheat spike actually has circadian nyctinosity, very similar to what I spoke about before, where the individual spikelets flex in and out as the day and night cycles turn in. And the wheat spike is mostly unexplored in terms of photosynthesis. If you type in spike photosynthesis into Web of Knowledge, you get about 51 manuscripts. If you type in wheat leaf photosynthesis, you get about 4,600. And this is for a couple of reasons. There's a common misconception that the spike only holds the grain and it passively accepts carbohydrates from the leaves. And that spike photosynthesis, even if it did occur, it doesn't significantly contribute to yield. However, a recent paper by Malero et al. Showed that spike photosynthesis could contribute up to 40% to final grain weight. So to put that in context, that could be a 40% improvement on top of, say, the 4% improvement we just saw looking in the field at nighttime temperatures. Another reason that spike photosynthesis has not received very much love over the last few years is the techniques to measure it don't exist. As a complex 3D structure, you really need a lot of infrastructure and the technology to measure it. If you compare that to a leaf, a relatively 2D organ, we have a lot of devices that measure flat leaves. We don't have very many devices that measure 3D structures. So why now? So climate change is accelerating, our heat waves are becoming longer, hotter, and our demand for novel targets to improve the climate resilience of our crops is really pushed forward. We need to improve it not just for food security in response to climate change, but economic security as well, as different countries experience different amounts of climate change. At the project scale, there's also advances in technology. So there's improved capability to quantify the physical differences of the 3D spike. In this project, I'm using X-ray CT imaging to now enable computational fluid dynamic modeling of the spike. And finally, I'm bringing in my own advances in chlorophyll fluorescence imaging. I've developed several high throughput imaging techniques now for looking at leaf heat tolerance, and I want to apply those to the spike. So the project has 4 sections. The first section is to develop a high throughput phenotyping rig, which enables looking at spike heat tolerance. And here you can see what I think is the first image of spike heat tolerance. So at 34 degrees, you have 2 genotypes. They have very similar efficiency. And at 47 degrees, genotype number 1 has a higher photosynthetic efficiency than genotype number 2. Once we have this set up, we can screen high numbers of spikes for heat tolerance, and that lends itself to genetic studies, so looking at the underpinning genetic variation of spike photosynthetic heat tolerance. I'm utilising contacts at Summit for particular populations of wheat that I think have the most impact, and also contacts at Erlen Institute to look at marker trait associations that might be linked to spike heat tolerance. And once we have those, it increases our impact of finding out these differences in heat tolerance. Once we've been able to look at large-scale population, I really want to narrow down and explore how the form of the spike influences the function. And for this, I'm using the Hansfield facility at the University of Nottingham, and they produce these beautiful 3D scans of wheat ears. So not only does that allow you to twist and change the 3D shape of the spike in space, but it also allows you to take images through the spike, which allow you to look at the physical structure as it changes and as the spike matures. And then finally, I want to translate this into the field. It's a very ambitious task for 3 years, but basically what we want to do is to work with the Wheat Research Centre at Nottingham. It's one of only 2 in the world that specialise in introducing wheat wild relatives into modern germplasm to improve genetic diversity. And in this case, I want to be able to suggest wheat wild relatives that might be best used to improve spike photosynthetic heat tolerance, so that pre-building material can be made to go to Summit to start the pipeline of having impact in the field. So that was my talk. I'm sure it was a bit faster than maybe intended, but I'm very happy to answer questions that anyone has.
Thanks for that, Lorna. That was a lot of data. That was packed, and your fellowship looks like you've got a lot to do. Robbie, you're on mute.
Yeah, I put a question in the chat, Derek. If you want to just read it, it's fine.
Okay, no, you go ahead. You're there. Just you ask.
I was just wondering whether there was any, or whether you even looked at any compositional differences or morphological or developmental differences between the cultivars that you used in your panel, particularly the panel that was looking at differences in or variation in not stomatal conductance. What do you call it now? What did you call it?
Nocturnal.
Cuticular conductance at night time.
So whether we looked at the composition, so for the phenology, we did assess phenology, so that did play on our minds, so how long it took to get to key growth stages. And although the phenology was slightly faster in the heat tolerance, you're only talking about 1 or 2 days faster, because we're only increasing it by 2 degrees as such. So those differences, they weren't so big. In the growth rim, there were differences in phenology, for example, with the Watkins lines and our modern elites, because the Watkins lines are a little bit of a wild card. They do tend to like to flower late compared to the elites, and they produce a lot more biomass. So they were different. For the cuticular conductance, we haven't assessed like composition, or for example, what would be higher in my priority list would be looking at, for example, membrane thermostability when they're younger, so how tolerant they are being disrupted by heat. And that would be really interesting. I also think it would be really interesting to assess, for example, like the lipid composition of those leaves, and see if you have particular genotypes that maybe have more waxy leaves when they're younger, are they more or less susceptible to heat? Is that a greater cost? So do they take longer then to achieve maturity and things like that? That would be really interesting.
So did you notice whether or not any of the accessions were considerably more glaucous or glossy than the others?
So yes, but you only really notice that when they get to the grain filling stage. It's very obvious. It's one of those differences that you do need like the field for, I would say, because I would think in the field in Mexico, glaucousness is much more obvious between different genotypes, just simply because of the extremes of heat at that end grain filling period. In the growth room, not much differences were noted, but it's something that maybe we could use like reflectance indices for, because there's several reflectance indices now that you can use to assess glaucousness, which would be really interesting as well. When we did the cuticular conductance, it's worth noting that we only did tillering, so before they hit flag leaf, and booting. So we only did those two younger periods because I was just interested to see if they changed, and they do. So it's a good assessment, even if it's a very small amount of water.
Thank you.
Okay. Sarah, you've got a question too. Do you just want to ask Lorna directly?
Okay, I don't know if you can hear me well, but
Yeah, you're fine.
I'll just read out what I typed. So thanks for a really awesome talk. That was super cool. I was really interested in the differences you saw in nocturnal stomatal conductance between the ab and adaxial surfaces on your leaf, and also between your genotypes. So I know you presented data suggesting that it wasn't, you don't think it's due to differences in stomatal density. So I'm curious, what your explanations for the differences, especially between leaf surfaces, could be. So, other variables, like, do you think the stomata themselves are behaving differently kinetically? Do you think other leaf features, so Robbie's already mentioned cuticular wax, but for example, leaves, or not leaves, leaf hairs, for example, influencing kind of the microenvironment around the stomata, so that the stomata themselves are kind of reacting slightly differently, or something else entirely? What are your thoughts?
And so it is really interesting because on the outset, there doesn't seem to be very much of a reason for there to be variation. But if you look a bit closer at the data, there are some genotypes that seem to have greater magnitudes. So, for example, Sienna has a Durham wheat, so that's more of a Mediterranean grown wheat. And it might be linked, for example, to water availability, which is why we're doing that drought study. Durham wheats tend to have very big differences in water availability. So they'll have like high water availability, then suddenly very low for a long amount of time, and then if they're not irrigated. So it might be a kind of a compensatory thing. So if, for example, they're well irrigated, they just maximize that condition. They're going to have their stomata open a lot more than, say, a fully Mexican elite line like Pavon 76 is a bread wheat that was developed in 1976. Fantastic variety. It's still very high yielding, but has a very, very low nocturnal conductance and a high heat tolerance. So I'm not sure whether it would benefit from having high nighttime conductance.
So are you suggesting that it is stomatal behavior, so it's just whether or not these genotypes are opening or closing their stomata, so it's finding differences in that response. So are you going to measure that, or?
So we started doing some of the measurements. This is more of a logistics question. So when we go to the field, we get maximum 2 hours, maybe 1 hour before we have to stop measuring. So we have to do it dusk. We can't do it at dawn because the plants are already soaking. But one of the interesting things that a lot of people say about nocturnal stomatal conductance is that anticipatory response to it. So if you have high nocturnal conductance,
Uh-huh.
you're available to open rapidly in that period of high humidity at dawn, so that you lose less water, but you can gain carbon. So I did check that for our growth room, because our growth room, you can't spray dew in a growth room, it won't like it very much. So I did have the ability to look at dawn transitions. So and we did see that those that were open more pre-dawn were able to open faster.
Right.
Dawn had occurred. But I don't think that's all of the story, because there's something that, one of the things that I thought was probably one of the things I've come across multiple times with stomata is roots. And assessing those in the field is very, very difficult. And one of the things I think is really interesting is if you're growing in a field or an irrigated condition, if it's a flood irrigated or a drip irrigated condition, you get very different root physiology. And that root physiology might also link to the stomatal ability to respond.
That's really interesting. Yeah.
You might have issues that way as well. So you might be looking at a twofold set of variation that causes that variation. That makes sense.
Right. And I guess the variation in what the signal from root to shoot might be to influence that behavior, or do you have a good idea about how that works already?
So not really. There's some really interesting things. So we just put in a paper on this data. And so there's a couple of things that, for example, if you have more shallow roots, you might produce more ABA. You also, ABA might also not just be linked to stomata, so the ABA would shut the stomata, but ABA would also be linked to pollen development, which is a really sensitive period of when plants at night are heated. And a lot of the papers that explore nighttime processes in crops put it down to increasing the temperature at the point at which the pollen is viable, and that makes the pollen less viable, and therefore you get less seed. Not necessarily the water, but then the water could be the thing that determines the development of that pollen too. So it's really exciting, but it's also very difficult to dig up plants in the field. So there's lots of questions going on.
Interesting. Thank you so much.
Okay, it's interesting. A reductionist approach to science isn't going to solve this. You'll be dealing with so many different systems in the one. So you've stimulated more questions. So I'll give you a couple, some simpler ones, but maybe difficult answers. Luke was asking, was the genotypic difference in nocturnal conductance related to the variety's origin? It's a kind of pedigree approach.
It's a good question. So I don't think we could confidently say that with 12 genotypes.
No.
So on a premise, I would think that there would be some indication that a genotype that was developed under a particular availability or lack of availability of water, potentially that might be linked to it, but I don't think we can answer it with the number that we've done. But it might be a possibility. It might be that more Mediterranean wheats have higher nocturnal conductance. And remember, we didn't cover any winter wheats, because obviously Mexico doesn't really grow winter wheat. So yes, I would like to say tentatively, there's plenty of variation out there, so there's no reason to say that it couldn't be environment specific to where it was developed, but you'd need a global panel.
Yeah, okay. So the other one was probably more to do with the work you're starting to develop now. Is have you got an estimation of the contribution of spike photosynthesis to yield? Because I think those numbers you gave are mind-blowing.
Yeah, so the papers I really pointed towards the Malera et al. Paper, which was 2020. It's anywhere between 20 to 40%. And I imagine that changes with the duration of the spike. So something that, I forget it as well, but the spike is actually available on the plant and in the atmosphere, so exposed to light for over half the duration the plant is grown. And yet we know very little about its contribution, its quantifiable amount of CO2 uptake or release, because obviously as it's developing and producing grains, it's going to increase respiration. And at the moment, we can estimate that. So a lot of gas exchange you can base on a flux basis, so how much CO2 exchanged, but we can't estimate stomatal conductance or CO2 assimilation because we're lacking some of the data that's required to estimate those parameters. So for a leaf, we'd have like boundary layer, pathways of resistance, all of those things that would allow you to estimate an actual CO2 assimilation, we don't have those. So I'm really excited to build that up and to really put forward the tools. It's going to start with wheat, but I really want the tools to be applicable so someone can, I don't know, scan a tomato and use that to calculate a boundary layer to then put towards using it for gas exchange. Custom gas exchange setups becoming more common again. Lycor has a setup for a custom exchange chamber, for example, but with that comes the responsibility of accurately assessing carbon uptake and carbon uptake in response to stress, which is really exciting.
Kind of leads into what we're going to be doing at the APGC, so lots of that will be doable, I think, in our new toys soon.
Brilliant.
So I think, Raoul, you want to ask the final question? Are you there? I'll ask you then. So Raoul was asking, your night experiments would be equivalent to a heat wave or prolonged exposure to heat during the days. Heat wave, yeah. Would you expect the same physiological responses in both scenarios, like a heat wave or prolonged day exposure, to be the same? Or do you think different fundamental biological mechanisms are active there?
So this is another one of those really interesting questions that I can say a lot about, but I'm very frustrated that I can't answer immediately. So, so the experiment that we did was a prolonged heat exposure. So from booting all the way to senescence is an absurdly long nocturnal heat wave. Okay, so the chances of us getting acclimation there is huge, right? Because they have plenty of time to acclimate to warmer nighttime temperatures, which is interesting in itself. So then you ask the question, is it more useful to invest in a long-term acclimation that results in minimal yield losses, or is it better to invest in short-term rapid acclimation strategies that allow you to survive those short, sharp heat waves? From the initial data that I did, so I took some tillers from the field, put them in a dark room and measured the leaves at different temperatures. The flag leaf of these plants was acclimated to the different temperature within 4 days of the heaters being turned on. Now, for me, that's an incredibly fast amount of time. It's a very small increase in temperature, but it's incredibly fast. But some of the data, especially the ratio of assimilation to respiration, that shows us that there's genotypic variation there as well. So not only in how they're acclimating, how fast they take to acclimate, those kind of assessments maybe need to be taken more into consideration when trying to decide on germplasm that best responds to heat waves. I don't know if that answers the question well enough, but I think, yeah.
I think it's gone some way towards it. I think what it identifies is we're probably a long way off breeding in this area.
A long way, but I'm very positive that, you know, we can only do better with more information. Even if it's just to highlight that this could be taken into consideration. Most of the breeding profiles for wheat, especially from CIMMYT, they take into account response to heat waves, so late sowing. We don't take into account nocturnal heat tolerance at all. And it makes me wonder, would our models be more accurate if we took into account some kind of nocturnal heat tolerance? Although I can imagine I wouldn't be very popular saying that because nobody wants to go out there at 9 o'clock at night and assess these things. But it's an interesting discussion to have because there might be processes that maybe make the plant better able to respond to daytime heat stresses. We don't know yet.
I think it would go back to the old maxim, if you don't measure it, you can't manage it.
Yes, very true.
Kind of works for everything, I would suggest. So I suspect you probably will have to do that. Okay, I think we've grilled you long enough and we'll draw this to a close. I'd just like to thank you, Lorna, for what's been a fascinating talk and the first joint one between APGC and IBH. So everyone can show their appreciation in the normal ways. Thank you very much. I'm sure we will be working together soon by the sounds of it.
Yes, definitely. I'm very happy to chat to people about collaborations, anything, just generally, I'm very open to discussions about how we can push this forward. So I think it's really exciting.
Thanks very much.
Hey,
Bye, everyone.
Bye,
Thanks,
everyone.
Lorna. Bye.
Bye.